Welcome to today's Explainer. We are diving into one of the most massive, absolutely fascinating scientific mysteries of our time. What actually determines the absolute limit of our lifespan? Today, we're unpacking some groundbreaking new research that is going to completely change how we think about the fundamental biology of growing older.
Okay, let's dive into this. For a really long time, the secret to aging was thought to be a simple matter of which genes are turned on or off, like flipping a light switch. But it turns out the true answer isn't just about flipping genetic switches. The reality is far more intricate and honestly, way more fascinating than that.
Section 1. A new key to lifespan. We are looking at a true paradigm shift in longevity. So, the crucial point is how science is dramatically shifting its focus here. The traditional view focuses heavily on simple gene activity, essentially just measuring how much a gene is turned on or expressed, like adjusting a volume dial. But this new discovery, it focuses on gene editing, specifically how our cells actively alter, cut, and rewrite those genetic instructions through a process called alternative splicing.
Which brings us to Section 2. What exactly is this gene editing function, and how does it change our cellular instructions?
Well, it comes down to a biological process called alternative splicing. Think of it like this. After a gene is copied, your cellular machinery steps in and basically acts like a film editor. It actively selects which gene segments, which biologists call exons, to keep and which to just cut out and toss away. It does this to build a highly customized final instruction set. This editing process is exactly what creates unique biological variations known as isoforms. And this brilliantly illustrates the concept using an analogy we all know and love, Lego blocks. Think of those individual exons as single Lego pieces. By combining the exact same basic box in slightly different ways, you can build entirely different structures, right? A car, a spaceship, a tiny house. Your cells do exactly this. By splicing together different parts of the very same gene, they can create proteins with completely unique shapes and functions. And the sheer scale of this mechanism inside your body right now is staggering. Up to 95% of multi-part human genes undergo this exact alternative splicing process. It's a fundamental pillar of our biology. To put that into perspective, errors in this editing process account for about 15% of all genetic diseases and cancers. It is absolutely crucial. Let's move to Section 3, a comparative study of 26 mammalian species. It's time to map the biology. To figure out if this splicing really is the hidden key to aging, scientists conducted a vast, ambitious comparative study across 26 distinct mammalian species. We're talking about mapping a huge range of animals with maximum lifespans ranging from just over 2 years all the way up to an impressive 37 years. And to make this study truly comprehensive, they didn't just look at one random part of the body. They systematically mapped the splicing patterns across 6 specific organs: the brain, the heart, the kidneys, the liver, the lungs, and the skin, the whole shebang. And the data from all those organs revealed a massive breakthrough. They discovered exactly 731 specific splicing events that directly and consistently correlate with a species' maximum lifespan. This definitively proves that lifespan-related genetic editing isn't just random cellular noise, it's highly precise, it's predictable, and it's measurable. Now, what's really interesting about this is that standard gene activity and this alternative splicing process operate completely in parallel. They are totally separate regulatory mechanisms. This means that standard scientific tests, the ones that only look at gene activity, are completely missing this independent, vital layer of lifespan information. Which leads us perfectly into Section 4, the brain's special program. Let's look at the brain's unique rules. Because here comes the major plot twist. For standard organs like your liver, lungs, or heart, splicing patterns are tightly linked to the physical size and body weight of the animal. But the brain? The brain completely breaks this biological rule. It ignores body weight entirely and operates on its own isolated program that is exclusively linked to the lifespan of the species. Think about this staggering statistic for a second. There are 2 to 3 times more lifespan-related splicing events happening exclusively in the brain compared to literally any other organ in the body. The brain's unique editing program is just hyper-focused on longevity.
So what are these edits actually doing up there? Let's move on and see how this builds into real function. Over 15% of these major brain splicing events directly control vital cellular functions. We're talking connecting neurons together, building nerve fibers, and releasing chemical signals. These targeted specific edits directly dictate how our brain cells communicate and adapt to stress over a long, long lifetime.
Finally, Section 5, a future map for extending health span, the future of aging interventions.
So what does this all mean for you and me? Why does this molecular biology matter? Well, to be perfectly clear, this research isn't some overnight magic cure for aging. Instead, I want you to think of it as a treasure map. As we increasingly live into our 80s and 90s, often battling chronic diseases, this map gives scientists brand new targets. It points toward future medical interventions designed not just to extend the sheer length of our lives, but to maintain our cells' adaptability. It's about extending our healthy, active years.
Which brings us to our final thought for today. If aging isn't just about genes blindly turning on or off, but rather how those cellular messages are actively edited, can we eventually learn to guide our own cellular editing? Could we one day use this biological map to completely redefine the very concept of aging itself? It's a truly fascinating frontier, and I hope this leaves you incredibly curious to learn more about the future of our biology. Thanks for joining me.